First, I would like to thank the organizers for the opportunity to present our data. We actually have, over the years, developed a multivalent vaccine platform. And we have developed a number of targets against a number of diseases that are not only extremely pathogenic, also has multiple subtypes. So what I'm going to talk about as an example today is a tetravalent dengue vaccine. So dengue disease is caused by 4 viruses, actually 4 types of viruses as the dengue viruses. This type of disease infection of dengue virus causing so-called breakbone fevers because the symptoms are so severe. Sometimes the patients often say, I'd rather die instead of having it a second time. And dengue infection can cause hemorrhagic fever, dengue shock syndrome, dengue encephalitis, and of course, the death rate is about 2.5 to 5%. There are 4 types of dengue viruses. Actually, there's no cross protection. So therefore, a patient not only can have 1 time, but it can have 4 times infection with the increasing severity because the immune enhancement of the disease actually not a protection. So of course, we are working with the Department of Defense. Dengue epidemic or dengue infection can be extremely debilitating to a military. So the number of cases dengue has increased almost exponentially. And recent data suggesting there's not only 200 million cases, actually it's over 1 million cases, actually severe cases require hospitalization. And recently, of course, dengue has been spreading all over the world. 2/3 of the population live in dengue-infested area, but recently has been spreading very close to United States, actually currently in the United States, especially in Texas and Florida. And I was in a meeting in Puerto Rico, just reminded, actually Puerto Rico is part of the United States. Actually, there's several hundred, I can't remember, the tens of thousands cases annually, actually with 2.5% to 5% death rate in Puerto Rico. So it is a problem. It is also a problem within the United States. So there's many challenges in developing vaccines against dengue viruses. There has been attempt for the last 50 to 60 years, but unfortunately, there's still no clinical approved vaccine not available. So there are several vaccine candidates. Actually, most of the leading candidates is conducted by Sanofi Pasteur based on the chimeric virus between yellow fever and dengue viruses, a mix of 4 viruses together and trying to induce balanced immune response. The current leading candidates not only take multiple vaccinations up to 4 doses over 12 months period of time, make it extremely impractical if you actually have to vaccinate a third world country, even travelers vaccine. So the major challenge is you have to induce neutralizing immune response against the virus. So all viruses likely to require neutralizing immune response, block the entry of the virus to prevent the virus further spreading. So the non-steroid immunity has been proven not effective in multiple cases. Most importantly, you have to induce balanced immune response against all 4 serotypes. So weak immunity against one serotype not only do not cause protection, but it causes a phenomenon so-called immune-enhanced disease. Because some of the antibody can increase the infectivity. Worst of all is that without the neutralizing immune response, the virus can keep infecting new host cells, and CTL response can come in to destroy more host cells, but actually enhance the disease, destruction of host cells, instead of preventing the disease. So, and also most of the current vaccine is based on live attenuated virus, and you had to mixing 4 replicating viruses together, and considering the virus is an RNA virus, not only physically unstable, also genetically unstable. So you put 4 viruses together and replicating in the human body, it's unavoidable. The virus will recombine, will adapt, will mutate, that potentially generate more virulent viruses. In the case if you're using chimeric viruses, you have, in theory, you can potentially creating a new serotype of viruses. For example, if you use the backbone of yellow fever viruses, at least in theory, you can create a 4 serotype yellow fever virus. So actually that's, does have its safety concerns. So in order to induce neutralizing immune response, the first thing is you have to have the naturally conformed the amyloid protein. That is to induce type-specific neutralizing immune response. That's provide protection, actually cross-immune response actually enhance the disease. The secondly is induce balanced immune response. This easy to say, but it has proven to be most challenged. Not only the 4 antigenicities of the 4 serotypes have different strengths, different level of antigenicity. When you put them together, actually the replication of the efficiency of each attenuated viruses also directly related to the immunogenicity. If a virus replicating more efficiently will generate more immunogenicity than the virus replicate less efficiently. Furthermore, when you put 4 together, actually there is a so-called immune dominance. Not only dominate the immune response, but sometimes the virus can dominate the other types. So actually make the other type even less replication efficient or less immunogenic. So actually that created a major problem for dengue, development dengue vaccine. In order to induce neutralizing immune response, the first is the amyloid protein has to be in a natural conformation. One requirement is fully glycosylated, properly folded, and containing receptor binding site, which is the receptor bind to the binding site bind to the receptor that initiate the first step of virus infection. If you have antibody block the initial interaction, that's actually by definition the true neutralizing epitopes. So if you look at this, making the protein-based, epitope-based vaccine impractical because the glycoprotein is never linear. So it has a 3-dimensional structure. So this actually is a simplified infection pathway of flavivirus. So actually the several point is the glycoprotein, which on the right-hand side, the conformations keep changing, depends on the stage. It is completely protein complex with a 3-dimensional structure. So if you, let's say you take a ball of a strand of yarn, you fold it in the bowl. You stain a black spot on the ball of yarn. When you linearize it, where's the stain? They can be everywhere. If you fold it back, what's the chance you can fold it back to the original shape that has the black spot still on the surface? It's nearly impossible. So you have a protein go through the purification process, even after renaturing, to make it naturally conformed is nearly impossible. And secondly, if you make the protein in bacteria and in yeast, there's no glycosylation, folded differently, there's modification that's differently. So make it extremely difficult to induce immune enhancement. The neutralizing immune response. Secondly, I do not have the diagram here, but actually this is the budding pathway. But if you can reverse it, you can also consider it as infection pathway. So the glycoprotein actually 3-dimensional, you had to block the infection at the entry site. Also, if you reverse during the budding process, the antigen associated with the cell membrane, this is the most strong form of immune stimulation. Also, this process doesn't last a few hours, lasts for days, until even weeks, until the immune response being fully activated. So naturally, if you're infected by dengue virus, you suffered it through, you survived. You're immune to the specific type dengue virus, but not the different types. So that suggests with the natural infection, you can develop natural immunity against dengue virus. If you can achieve the same without having the infection, you can develop an effective immune response. If you can achieve the same against all 4 serotypes, but without the disease, you potentially can have a therapeutic dengue vaccine. So this is our strategy. We use a sheep in the clothing of a wolf, basically mimicking infection with a 3-dimensional antigenic information instead of linear, instead of epitope, to really present the infectivity in a natural form. So basically, we're using benign carrier to carrying the antigen, the genes encoding the antigens into the cells to mimicking infection, have a cell look like infected. So they are the sheep in the clothing wolf. We can create a sheep in clothing of a tiger. We can create a sheep in the clothing of lion. But inside is not crippled lion, is not crippled tiger, is not crippled elephant, is a sheep. So the attenuated virus is a good approach. But if you cripple a wolf, as you fight, you might win. If you cripple a lion, a crippled tiger, the chance you win the battle has become remote. And there's also a chance the tiger will recover. So in this approach, inside is a benign carrier, can never recover, can never become virulent. So that's basically our approach. So we use a complex endovirus vector system, not be confused with first generation endovirus vector system. It's a vector packed into a shell of endovirus that can accommodate up to 6 antigens. So we can create a vaccine with multi-antigens. The antigens induce CTL response, antigens induce neutralizing immune response. In addition, for the antigens that are weakly immunogenic, we can also allow the vector to create cytokines or immune enhancement to actually further mimicking a natural infection. So the cells, once infected, they secrete stress signals, which also enhance immunity. So in this case, we can not only present one type of antigen, we can create the entire repertoire of the antigenicity. For example, if we use for HIV, we can allow the cells to secrete empty viral-like particles, which is empty viral particle like HIV. Not only has the cell-associated phase of immune stimulation, but it also has the free particle to stimulate the immune response. So it can be mimicking the entire infection pathway, but there's no virus to replicate, no chance to induce disease. So that's all talk, but what's the evidence that works? So we heard the earlier talk about the dangers of Ebola virus. That Ebola, take no offenses, Ebola is more lethal than smallpox. They kill 98% infected. A single particle killed nearly 100% all primates, including human. So non-human primates is not a model. They get the same disease. And they're highly contagious. We used to believe it is not be able to transmit through air because that's just the imagination in the movie Outbreak. It recently has been not only been proven by previous Soviet Union, recently proved by US Army. Aerosolized Ebola virus in inhalation of one single particle causes massive hemorrhagic pneumonia. The normal pathway for infection is multi-way the entire internal organ. The liver, basically, the scientist said you take the liver basically flow through your finger. But for all airway infection, you have a massive pneumonia, hemorrhagic pneumonia, before actually the liver get affected. So the death is much quicker, even more lethal. So when we put the 3 or 4 Ebola Marburg antigens into our platform, with a single transduction, you can see the cells actually expressing the glycoproteins of Ebola virus, Marburg virus on the cell surface. The level of expression is much higher than real infection because there's no pathogenicity to the cells other than overproduction of the antigens. So these cells look to the immune system and heavily infected by Ebola viruses, while there's no Ebola viruses at all. So these are the sheep in the clothing of a wolf to stimulate the immune response. So the vector is quite efficient. In this case, it's GFP. We inject in the muscle. Under dissecting scope, you can see the entire piece of muscle become brightly fluorescent. If you imagine these are the antigens of any viruses synthesized in muscle, including HIV. HIV do not infect muscle cells. If you're expressing HIV envelope, in this case, a dengue envelope, the muscle looks to the immune system heavily infected. So therefore, you can imagine the immune system will mounting a full spectrum immune response against dengue viruses. So in this case, actually tested by US Army against 6 types Ebola and Marburg viruses, each challenged at 1,000 times the lethal dose. The requirement is 100% protection. So I'm going to only show you one example. In this case is Marburg virus CS67. So with a single vaccination, the immune response goes to maximum, reach the ceiling, reach the plateau. So with the challenge at 1,000 times the lethal dose, in this case will be Zaire Ebola virus at 1,000 times the lethal dose. All non-human primates survived. Not only that, the antibody level did not increase, demonstrate the antibody reached the maximum level. And the same group of monkey challenged the second time with the Marburg viruses. And again, 100% protection, there's no increase in antibody level. So demonstrate again, with a single vaccination, the immune response already reached the maximum level. I can repeat, show you the same slides 6 times. There are 6 types Ebola and Marburg viruses through the challenge experiment. So it's not only that, it's the same group of monkey received 1,000 times the lethal dose of different viruses at a different time. Almost like a bulletproof vest, you want to shoot in the same spot 6 times, and it still require the protection. So this is the vaccine has done. So basically, we have a serious experiment to show not only can protect against aerosol challenge in non-human primates, and also in the presence of pre-existing immunity against AD5, because the shell is still AD5, there is a big argument whether the pre-existing immunity can prevent a vaccine from working. Of course, there's plenty of data to show, yes, it does not, the vaccine still works, but does not prevent the vaccine from working. So actually, this is the first generation of our dengue vaccine, basically to assure the 4 serotypes are equivalent, expressed at equal level. So we actually, the first generation, we're using 2 complex antiviral vector system and using identical expression cassette put in a nearly identical genomic location, each one at either end. And we put 2 vaccines together, we can induce tetravalent immune response. In this case, have nothing to do with viral replication, and only have anything to do with the expression mechanism to drive the antigen expression. So if the antigen, because they are driven by identical promoters, the identical antigens, identical genomic location, so in the way, they're supposed to be producing identical or similar level of immune response. I will show some data actually consistent with the other experiment. The pre-M and E antigen of 4 dengue serotypes, actually the antigenicity is not equivalent among the 4 serotypes. But with this mechanism, the challenge experiment was done by US Navy at NMRC, Medical Research Center, Medical Center. Oops. So this is actually is the experiment approach. Basically, based on US Navy, you can only reduce the number of viremia based on the vaccination. You cannot completely block the viremia based on their challenge strengths, because they want to see the differences, they want to see the gradient, all right? Secondly, they want to see the long-term challenge, not only 3 months right after vaccination, they want to see 6 months challenge. And in the Navy experiment, they vaccinated twice, because they never seen, in any case, 1 vaccination can work for dengue viruses. So they vaccinated at 0 week, 8 weeks, and challenge at 3 months after vaccination. And the second group, the challenge vaccinated at 0 week, 8 weeks, and challenged at 6 months after vaccination. So these are the raw data, but I do not expect you can read it. I can't even read it here. But this is graphically, actually ignore the scale. Actually, just remember the highest is 1,200. So actually the scale is wrong. But the proportion is correct. So this is 28 weeks, 29 days after vaccination, and 85 days after vaccination. You can see actually induced the vaccination, 2 vaccination induced high-level immune response against all 4 serotypes. As I said earlier, I thought it would be in theory they're equal, but they're not. Because actually the immunogenicity of different serotypes are different. Even use identical promoter, identical cassette in identical location, the immunogenicity are not identical. If you complicate this mechanism with a difference in replication, that's why all the other systems, it's very hard to induce balanced immune response. But if you look at it closely, based on the information, large amount of data to show, actually there may be different groups have different strengths, suggest slightly differently. Dne3 has the weakest immunity. The second weakest is Dne4. And Dne1 and Dne2 have a similar immunity. So during this process, we actually did one genetic trick and Dne3, because it's the weakest immunity, the hardest to protect, and actually has been shown most pathogenic. So we actually codon optimized Dne3, not the others. If you look at the immune response, actually Dne3 become the highest. The second lowest, which is Dne4, remain to be lowest in terms of immune response. So actually, because the antigenicity of the protein naturally have a differences, but we can use a molecular approach to enhance immunity. So we actually enhanced the weakest, Dne3 makes it the strongest. So actually that suggests the best way to do, seems the order is it. The best way to do is to enhance the immunogenicity of the others. Actually, the day after vaccination, this is the viremia. Under very stringent challenge conditions done by US Navy, based on their words, they never seen complete protection before. Actually, Dne1 and Dne3 now complete protected. Dne2 have one monkey has less than a day viremia, but Dne4 has nearly 1 day viremia at 3 months challenge, and at 6 months, actually nearly 2 days viremia as after challenge. So this is under highly stringent conditions. Actually, the Navy has never seen before that's complete block viremia under such conditions. But nevertheless, if you can look at the level of immune response, Dne1, Dne3 are highest, Dne2 slightly lower, Dne4 is lowest. So this suggests we can improve the immunogenicity based on what we've done for the Dne3, which is codon optimized. So we actually codon optimized all 4 antigens. The reason is the dengue virus has to replicate both in human as well as in mosquitoes. So the codon is not optimized in neither system. So now we actually optimize the codon for human only. So it will have a highly expression in human, but not in mosquitoes. Can you do the same for attenuated virus? No. Because if you do so, you create highly pathogenic super dengue viruses that are extremely infectious to human. But in our case, because the virus doesn't replicate, it's a genetic vector to expressing the protein only as the antigen. So we actually codon optimize for all 4 antigens. After codon optimization, this is based on ELISA, you can see clearly again. With one vaccination, all 4 serotypes reach the maximum level of immune response at about 2 weeks, not 12 months. And the immune response actually maintained at a very high level. And this is a neutralizing immune response. And this time we can see neutralizing immune response as equally across board from Dne1, Dne2, Dne3, and Dne4. So this case, actually we can provide complete protection against all 4 serotypes. And in addition, because the promoter is ubiquitous, there's no species difference. There's no not influenced by genetic background. So the attenuated viruses tend to be influenced, depends what genetic background, you're either Caucasian or Asian. So they tend to, some of the vaccine trial actually failed because after safety trial, you take in a different genetic background, actually the vaccine causes disease. And is the vaccine safe? Of course, actually, the first generation vector has been in thousands of patients, including the CEP trials, and also in gene therapy for cancers. Those patients under severe chemotherapy and radiation therapy, under immunosuppressed, none have side effects directly related to the vector. And also we've done GLP biological studies. What's more important, I was criticized as a little bit over-arrogant. We used a dose 1,000 times in the New Zealand white rabbit this big, compared to what's the highest dose even for Ebola in human. FDA told us it's not necessary if you cause side effects and you have a lot of explanation to do, you have to retract the whole experiment and show there's a baseline. Retrospectively, I will never do it again, but fortunately, at 1,000 times the highest dose, intended dose in human, there's not a sign of any side effects in the GLP toxicology study, and of course in biodistribution. The vaccine is also very efficient to produce. We have developed a fully enclosed system with a single pass. From bioreactor to vialing, with a single pass, the whole system is enclosed, only taking samples out of the system. This just shows you how the vaccine efficiently produced in mammalian cells. So this is total biological mass. This is the mass of the vaccine. Relatively speaking, it's very, very high in terms of the vaccine production. With a single pass, we can generate the purity of the vaccine that meet the requirement for clinical trials. And the vaccine also versatile in several different ways. One is not only applicable to different vaccines, and also can be used in different routes, intranasal, oral, capsule, as well as skin patch. In this case, you do not need injection. Basically, put the patch on the skin, you get vaccinated. So actually, we have a lot of data. This is actually another vaccine based on the Spanish flu vaccine virus. Actually, it's a multivalent flu influenza vaccine against H5N1, including a Spanish flu. Spanish flu is BL4, so you can't use it for any other system to develop the vaccine. But our system can be used for Ebola Marburg, therefore also can be used for Spanish flu. This actually, the red is oral vaccination, and the blue is IP vaccination, intraperitoneal, which is the most efficient vaccine in the murine model, but not in human. The equivalent to intramuscular vaccination, and this is intranasal vaccination. So you can take oral, intranasal. We also have a data, actually you can slightly abbreviation, either sandpaper actually damage the superficial skin, you can get equal vaccination through the skin. The reason, as we heard the early presentations, most of the dendritic cells are concentrated in the skin in mucosal membrane. That's where the invasion of infectious disease started. So the skin and mucosal surface are the best vaccination site, not deep in the muscle. We actually currently completed the preclinical studies. Actually, there's some reason this is wrong. We completed preclinical studies. Actually, for some reason, there's the wrong slides. We've done all preclinical studies and the GLP toxicology study. We had a pre-IND meeting with FDA, and we are so encouraged. Actually, they were telling us once we've done the clinical trials, we get a certificate fast track. Not only the vaccine itself can be fast tracked, we get a certificate fast track that can be used, sellable, transferable to any other pharmaceutical products. Fast track another product doesn't qualify for fast track. So that indicating the need for dengue for vaccine. Actually, for some reason, this is not the right presentation. We also built a 50,000 square foot GMP facility, and we are just about ready to, once again, the CEO will actually start getting the approval process. Of course, the work is done in collaboration with NIAID, funded by NIAID, NMRC, USAMRIID, Medical University of South Carolina, UTMB, and Southern Research. The biosafety was done by a company used to call Bridge Tech, and I think now they changed the name. Thank you very much.